Great. Thank you for your introduction, Elenia, and for inviting me to speak here. Yeah, exactly. So I will ask the question, where are we right now using AI in drug discovery? I mean, AI is a term that's in everyone's mouth and it's used in various areas. There's a lot of hype behind that as well. But my point is that the data we have to apply our algorithms is very different in different fields. Yeah, that's kind of the key message, as I will outline on the next few slides. And in the drug discovery context, that data is actually very difficult to deal with. And so I'm aiming to step back a little bit from that hype and to look at the situation of applying AI in the drug discovery context and to work out or to figure out how the data we have in that area differs from other areas. And so any statements, as Elena said, of a company as well as an academic affiliation, any statements are made in my capacity as an academic and as a researcher here. So where are we right now? So we're right now in the third wave of computers in drug discovery. There was one in the 80s. So that's a fortune cover from 1981. That was the year when Silicon Graphics was founded, for example. That was also the year when the first DOC algorithm was published. It wasn't called DOC back then. And you see here, that's the next industrial revolution that was proclaimed, designing drugs by computer at Merck. And in those times, computer graphics came up, and that was quite a lot of structural work that was going on, visualization, structural work, structural modeling of proteins, for example. And then in the 2000s, we had another wave of computers in drug discovery. So that was more the bioinformatics wave, cheminformatics. The human genome was sequenced. So that was much more informatics or data-driven already. And of course, over the last couple of years, we have headlines that look similar. So how artificial intelligence is changing drug discovery and so on. The headlines look similar. Are we in a similar situation? Well, maybe to an extent, but certainly some aspects certainly differ. We have much more data, for example. Our data is better annotated. We have ontologies, for example. We have very, very powerful algorithms, different types of readouts, and so on. So certain things might sound similar, certain things change. And so I will try to work out what are actually the differences and where are we right now using AI in the drug discovery context. And there's a particular reason for that, because I'm old enough to remember, for example, the 2000 Biotech Bubble and the promises that were made in those times. And not everything that is promised at a given point in time becomes true later. So around 2000, the draft sequence of the human genome was published as well. And let's look at the papers at that time. So for example, it was claimed that the number of drug targets will increase by at least 1 order of magnitude, and target validation will become a high throughput process, and there will be more drug targets, so 3 to 10,000 targets compared with at the time 483. But what happened, if we look at more recent reviews, we see that the number of drug targets is only around 667 a couple of years ago. So that means that all those expectations, they weren't really fulfilled at least 16 years later. But of course, we can now target potentially mRNA. I mean, there were some very nice papers from Cambridge, for example, recently. We have different modalities. So we have ASOs, we have antibodies, and so on. So we have different ways of modulating biology. But still, I mean, that original expectation of the number of drug targets increasing, that wasn't really true. On the other hand, we can catalog biology much better. I still remember the times when people expected humans have 200,000 or so genes, and eventually it turned out that number is much lower, maybe 23,000 genes. Of course, you have different splice variants and so on, and post-translational modifications. But still, I mean, we can now catalog much better, right? But some of those initial expectations probably didn't become true. And certain similar aspects probably will apply in the drug discovery context as well. So that's the context. Sometimes it's good to step back a little bit and see, bring some perspective to the application of algorithms in a given area. That's what I'm trying to do here. So this is the outline of my talk. I will talk about chemical and biological data. That's what I would call the flat earth view. So that's a simplified view on chemical and biological data. And that view can still be very useful in practice. You can use this flat earth view, and by that I mean simple labels, which don't really apply to biological data, but still simple labels that don't consider the conditionality of the data can be useful. So this is the first section of my talk. And the next one is then the round earth view. And that considers drug discovery data and its complexity and conditionality. And that's what I would call the elephant in the room, because right now many people talk about AI in drug discovery, but much less about the data we use to achieve those goals. But as I will point out, biology, pharmacology is very conditional. things depend on dose, for example, right? And if you don't consider all of that, then the conclusions you draw may be wrong. Dose really matters, and many other aspects matter as well that we often don't know. Yeah, and that chemical and biological data can be very complex, very conditional, and that's what I will point out on the second part of my talk. And eventually I will come to some key learnings, and those learnings will be about the data we have, and that's not always the data we need, as well as model validation and human biases when it comes to using AI in the drug discovery context. So that's a simple view of the world, linking chemistry, protein targets, and phenotypes. And all of those angles, so I'm a chemist by training, but when I started my postdoc at Novartis, I started working on high content screening data. And if you grow up with a certain education, yeah, then you're trying to understand the world in this particular way. But that doesn't really work in some situations. And it certainly didn't work for me when I, as a chemist, which is quite systematic in many aspects, started to analyze high content screening data, which is very high dimensional, yeah, very fuzzy. The question was, where's there's a signal, yeah? And I realized that different domains behave very differently. And I think it's very very important to have this integrated view on data from multiple angles. And so in this case here, that's the chemical angle, the mode of action angle, and the phenotypic angle. And this is the data we always integrated in my group in Cambridge. And all those angles are actually underpinned by data. So from molecular structure to the protein, we have hundreds of millions of bioactivity data points that link structures to thousands of protein targets. Then we can annotate proteins with pathways or genes with pathways that we somehow link to phenotypes. But of course, that's more human thinking, right? We like draws, need draws. That's one pathway, that's another. That's mechanistic, that's procedurally much more easy to deal with, yeah, than if you have networks that reconfigure themselves depending on gene expression levels and so on. So pathways is quite a simplified view on biology, but still we can organize genes in some ways, let's say, and links proteins to phenotypes. And finally, we also have links between structure and phenotypes. So that's phenotypic response data. Compound causes an adverse effect, compound kills a cell, and so on, right? So that's the data we were dealing with. And but this is the world is flat view. And why do I say that? We believe our labels. So we believe a compound, for example, binds to a target. Is that relevant in an in vivo situation? Well, only if the compound reaches the target tissue. Yeah? A compound causes an adverse effect. Is that true? Well, it depends on dose. It depends on the genotype, on the disease endotype, and so on, right? So, but the world is flat view precisely does not consider all of that. It just means we believe our labels, right? So we assign a single label to a particular compound and one of those interactions. And that is simplified, but it can still be useful. But why is it actually simplified? Because in many cases, we don't actually have directionality in our information. We don't have causality in there. Think about GWAS studies, right? So we do statistics of identifying particular variations in the genome with particular phenotypes. And quite often, if we have data, we don't really consider whether the time point or the biological setup or the concentration is relevant from one assay to the in vivo situation, right? So all that conditionality, the complexity of the assay setup is quite often lost, and we just label our data. Why do we do that? Because we need to do it for machine learning or AI in many instances, yeah? But it's a very simplified view on the data. I will give some examples later. And why is that the world is flat view, where we simply label molecules, for example, with targets? Why is that still useful? Because we can answer questions. And these particular questions are usually of three types. So the first type is, what is the reason upon treatment with a particular compound for a given effect? So in this particular case, we have a link between the chemistry as well as the phenotype, and we're predicting the mode of action. And the next question we can ask is, we can go back to chemistry and we can ask the question, which compound should we make to achieve an effect? So we want to modulate the phenotype, and we can design a pathway focused library, right, based on the data we have. We can say we don't screen 1 million compounds, we screen 1,000 compounds that target a particular pathway that is upregulated in a cancer. And the third question we can answer is, we can go back to the phenotype. That works very well, for example, in HIV. So I have a particular reverse transcriptase inhibitor. I can sequence the viral genome. It's very cheap right now as well. It's a standard to do that. And then I can say which patient actually responds to the application of which protease or reverse transcriptase inhibitor. That works very well in HIV, for example. It really makes it a manageable disease. And so it makes sense to integrate data of those types. And we can use it in concrete terms, for example, for target prediction. So in practice, we have data that links structures to phenotypes. For example, compounds to particular adverse reactions or to particular cellular effects, and then we can use that structure, and based on data, we can predict protein targets. Using fingerprints, for example, in machine learning models, you can do that in many different ways computationally. And one example here is from Astrazeneca, so where we used on the order of a few 100 million data points, and so that data is relatively rich, so it's millions of data points. It's across thousands of protein classes, but one of the big problems is it's very biased data. So you have some classes where you have 20 active compounds and very close analogs, another class 20 compounds, very diverse compounds, another class you have 10,000 actives, and some of them cluster in series, some are diverse, and so on. And so that's one of the problems. Yeah, you have very different data distributions among those classes. But what we can do with that is we can use that information and build machine learning models that predict based on a molecular structure which targets are likely to be hit by a particular compound. So this is what the two examples of compounds here on the left show. So these are, the left one is similar to sporine, it's a kinase inhibitor. So you see the targets that are predicted likely to be hit by that particular compound. And the right-hand side, it's Gleevec, so hits BCR-ABL, ABL1 in the standardized form. And so you can see that you can predict protein targets of compounds. That is very useful in practice. It considers the data you have. On the other hand, because you don't have complete data coverage, you're very likely to produce false negatives, right? Because you don't cover all areas of chemical space, not all areas of target space. So you use the data you have, that's good. But you need to be aware that if you have data-driven methods, false negatives are, of course, very frequent. Yeah? But in practice, that's useful. If you do high content screens, predicting the targets to understand the mode of action, that's of huge importance, for example, in pharmaceutical companies. So what is characteristic here? So in this particular case, machine learning models do work. Why is that? It's to a good extent because data have two characteristics. It's unfortunately not always true where you would like to use AI in drug discovery. You have quite a lot of data, and so it's relatively large scale. On the other hand, the data is relatively homogeneous. So what does it mean? If we have a target-based assay, the setup is, at least compared to in vivo data you can generate, relatively homogeneous. So if you have a 5 nanomolar compound against a particular kinase, even if you vary your assay setup slightly, you have a different ATP concentration or so, you're still quite likely that that compound has a certain activity, quite good affinity against a particular kinase. Yeah? And that variation is much smaller compared to in vivo data. So it's a relative statement. It's not that bioactivity assays are always entirely homogeneous, no, but compared to in vivo data, it's relatively more homogeneous how the data is generated, right? So you have lots of data and it's quite homogeneous, which means we can label our data, yeah, much more easily than in vivo data. I will give examples later. But of course, there are various problems with the data, data biases, for example, yeah, labels are still to an extent heterogeneous and so on. But still, on a relative scale, you have lots of data and quite homogeneous data. But of course, that was the world is flat view. But in reality, things don't look like that. If you think of pharmacology, for example, compounds are not active on a target, right? You have partial or full agonists, so allosteric modulators. You have different residence times. It really matters for GPCRs, for example. You have very different side effects. If you have different on and off rates, for example, different signaling cascades you can activate or inactivate in GPCRs and so on. Also labeling a disease, for example. How do you label a disease? You know? Do you use the mutation? So do you use it if you have a genetic disease at all? Do you label it on that level, or on a pathway level, or on a phenotypic level? And sometimes you have individuals that have mutations that actually mean they inherited a particular disease, but that might not be visible in all individuals on the phenotypic level, right? So how do you actually label even something that sounds as simple as a disease? And that might be easier in some areas than others. For example, CNS diseases, right? Depression or anxiety and so on, they are symptomatic clusters, yeah? So it's very difficult to label the data we have in many cases. So the point here is computer science is very powerful, but is our data? And quite often, I believe that is not really the case, due to the examples I've given here, and a few more will follow. And this is in some sense the most important slide of the presentation. So AI has been very powerful in the image domain and speech domain. And here's an example of the outlines why the domains differ. So you see here the image of a cat. So that's an object, that's the cat. It's represented in matrix form, and that's a very good representation of the object. So we can represent the cat in this picture in matrix form, people would recognize it as a cat, so that's a truthful representation. And this matrix representation can be captured in a model, convolutional network, for example, where you have various filters, edge detectors, and so on. And this model recognizes shapes, which is also the shapes we would recognize in order to recognize an object. So that integration is very smooth. And finally, the label we assign of a cat in that picture is something we can unconditionally assign. Sometimes the object, of course, might not be fully visible, partially hidden, and so on, but still, compared to chemical and biological data, that annotation is much less conditional or even unconditional, which is not true for other domains. So you can see that whole integration of representing the object, learning from the representation, and the labeling. There's a much more smooth integration in case of images. The same also for waveforms or for speech. In the chemical domain, things are much more tricky, right? So we have a molecule here, but the question is, how do you represent that molecule? So you can have different conformations, tautomers, protonation states. Maybe you have reactive groups. Maybe the compounds form aggregates, and so on and so on. So this is much more ambiguous how to represent that molecule. And then you need to transform that in some type of representation for the computer. And here you have various fingerprints, pharmacophores, and so on. And that's done usually by trial and error. There's a ton of different representations of molecules. The question is, which one should you use? And here, of course, what became popular is learning the representation from data. Yes, but there are two problems with that. One is the conditionality of the data. That's a conceptual problem. The second one is of relevant data. Quite often we don't have enough anyway. So we don't have enough data to learn that representation in the first place. So it does not really answer that question in vivo relevant areas. So you see already the representation question is much more difficult. When it comes to modeling, it's far more trial and error as well. There's no native modeling technique that we can use to learn from chemical data. And finally, the labels are much more conditional. So toxicity depends on dose, for example. Water, even water is toxic. If you drink 5 liters of water right now, or over the next half hour, you'll have severe problems, right? So the question is, how do I assign a label to a molecular structure? And this is certainly much more difficult than in case of having a cat in the picture or not. So one example of conditional labels, adverse reactions. So it's difficult to compile information about adverse reactions. You have lots of reporting biases and so on. But in computer science, sometimes you have studies that try to predict, does a drug cause an adverse reaction? Yes or no? And is that a real question you can ask? Well, in practice, that is actually much more difficult, right? Because it depends on the genotype, it depends on which dose you take, and then you might see adverse reactions in a certain fraction of cases, 30% of cases at different severities. Maybe you see that more frequently if you co-administer the drug with a drug from a different class, and maybe only in males and after 3 months of dosing with a particular dose, right? And so as you can see, the question of does a drug cause an adverse reaction is conditional based on dose and so on, endotype, genotype, right? So the labeling is very, very difficult. You cannot really do that. And quite often you don't have enough data to really fill all those pieces of information that are missing here. So it's very difficult to have data and figure out all the conditionalities of the data in the model. So either we neglect parts of the equation or we don't have enough data to really consider all of them. That's usually the two different problems we can choose from. So does a particular drug cause an adverse event? Well, it depends, right? So it's very difficult to label that. And there are usually two different domains of data in drug discovery: early discovery and efficacy and safety. So in early discovery, quite often you have simple readouts. We have large numbers of data points for training models, but it's proxy assays. So it's very early stage readouts, log P, log S, or something like that, or bioactivity against targets. But that is very early stage, less in vivo relevant. And then you have efficacy and safety assays. That is usually quantitative data. It's much more complex models to generate that data, so you have less of them. And you see the problem already, I assume, right? On that side, you have data that is very conditional, but you also have much less data because it's much more costly to generate efficacy and safety related data, right? And that is true also from the practical side. For example, if you do animal studies and you have a cage at the top or the bottom, at the bottom, animals have much more stress, right? So they react differently. If the handler is male, they're also more stressed, right? So all of those are confounding factors in your results. And so also from the practical side, it's more difficult to generate that data. So you see, you have early stage data that is less relevant for the in vivo situation and have more of that, or you have late stage data that is more relevant for the in vivo situation, but it's more conditional and you have much less of that, right? And that is a problem, of course, because we don't have the data we need to train in vivo relevant models in many cases. Unfortunately, the problem setting is exactly the other way around. So in early discovery, we have the easier question. So as a discovery setting, so like a virtual screening setting, where you're looking for any active compounds, right? That's an easy question to ask. There are many possible answers. I can pick from millions or billions of compounds. On the other hand, for efficacy or safety, we need to predict for a particular molecule, right? So we don't have that choice of a million molecules. No, we need to make a prediction for one molecule based on much less data and much more conditional data, right? And that is much more tricky to do. So you see, quite often we don't have the data actually to answer in vivo relevant questions, right? So there's a certain mismatch of the data we have and the data we need. Sometimes you see success stories of AI and drug discovery, and this is just a reminder, a ligand is not a drug. So we don't want to treat proteins, we want to treat humans. And so if a compound only has activity on a primary target, that is not really sufficient, right? Then you have a ligand. We have millions of ligands in PubChem, but we don't have millions of drugs on the market, right? So don't only look at activity on the primary target, and consider physical properties, PK, and so on. And you need to have efficacy and safety. Yeah? So it's not about proxy assays and drug discovery. It's about efficacy and safety in living organisms, be it humans or animals. Yeah? But it's about effects in the organism. And why is that important, actually, that we don't have the right data? Well, because the problem actually occurs in clinical phases in drug discovery. So here you see the different phases in drug discovery. And if we speed, quality, cost of decisions, how much money we could save on those projects. So speed in white, quality in black, and cost in gray, right? And you see, if you're able to reduce the cost of phases, you can save a little bit in lead optimization, right? Because you have many iterations of those cycles, and they can be costly. However, the real savings would come from having better compounds, so better quality, the black bars, in the clinical phases, right? Those bars are much higher. Why is that? Because clinical phases are costly, failure rates are high, and the phases are long, right? So the point is not for us basically to make faster decisions, yeah, or save money. I mean, that's all nice, but the real effect comes from having lower failure rates. And so from actually having better compounds that go into the clinic, right? But we don't have the right data to do that, unfortunately. So to have a few concluding slides here. So about data we have as well as model validation. So the data we have is not the data we need. So what do we mean by that? Proxy assays have been used in drug discovery only to be one input into decision making, but they've now evolved in the context of AI to models in their own right. But that is not necessarily the way they're meant to be used. So you have certain assays that are meant to indicate particular properties of compounds. For example, there's a Google mitotoxicity model, but it doesn't quite matter. It's particular proxy assays that are meant to be used to inform decision making, right? But right now, in the AI context, they are taken as models in their own right. So there's a model that is then meant to be used for decision making based on numerical output. But this is not really how proxy assays were meant to be used. Yeah? They're meant to inform human decision making. Yeah? So the data gets used in the wrong context, so to speak. And there are usually two problems with the particular data that can get generated. They're listed here at the bottom, and that's usually what I ask for right now. If there's experimental data of some type, is it hypothesis-free or hypothesis-driven? Yeah, hypothesis-free just means you generate lots of data, but you don't actually know if it's useful. Yeah, hypothesis-driven means is a targeted way of generating the right data for the right purpose. The other question is a technology push or science pull, right? Technology push means we can sequence, so we sequence everything. Science pull means genetic mutation tells us something about sensitivity, so we need to look for that mutation in the genome, right? It's a very different way of generating data. And this is a pyramid that illustrates where computer science or AI comes in science, right? And it's a pyramid that is based at the bottom on a question or hypothesis. And that gets used to generate the right data. That data needs to be put into the right representation and analyzed using the right methods. Yeah? But the method only comes on top. And that's where there's lots of attention right now in AI, computer science methods. But the methods only analyze the representation of a data that needs to come from a question or hypothesis, right? And if that base is not there, then the method cannot save anything that's not in the data, for example. Yeah? So you need to consider the data you use to answer a question from that bottom up to the top and generate the right data to answer a particular hypothesis. Model validation, model validation is difficult. There's only one sentence I want to say here. Model validation is always process validation. So if you consider the validation of AI models in drug discovery, that always depends on, for example, your virtual screening model, that always depends on which vendor library you pick from, right? You have a given hit rate and you compare your model to baseline, yeah? It always depends on which library do you pick from. There's always a human component. Don't forget that. Stephen Kearns from Google on our Relay, he was writing very nice papers on that. And to conclude here, so the bigger picture, what are problems with using AI in drug discovery? Of course, AI is in no small part where it is right now due to human psychology. Hype brings you money and fame. Realism is a bit boring, right? There's fear of missing out, and that is very prominent in big pharma, for example, who have long budget cycles and so on. And there's always the fear, oh, maybe there's a more nimble startup that actually has 5 million or 10 million of funding and can start tomorrow, right? So there's psychological factors. Beliefs quite often drive decision making. Maybe they really have the secret sauce. Everyone needs a winner. So after a company invested 10 million or so in something, you want to show success, right? And all of that leads to biased reporting of successes. So it's not really a scientific way to compare the benefit of methods, right? There's a certain, human beliefs or certain drivers that influence how results get presented. That's not always beneficial for science. Selective reporting of successes leads to everyone declaring victory, but in reality, actually, no one knows what's going on. That makes it difficult to really advance a field, of course. Yeah, what could make it need to be as possible to generate relevant data, right? And that might be omics data, but you need to be careful to extrapolate to the in vivo situation. Maybe it's cell morphology data, very cheap to generate. You see quite a lot on that, but we need to determine the applicability domain more. Not every readout tells you everything. Organ on a chip, yeah, possibly. The new workshop just 4 weeks ago, but you need to standardize organs on a chip much more, have reporting standards, right? Otherwise, you don't agree on what to measure and what to see in there, right? You cannot merge data either. So it's tricky, but you need to be in the middle, yeah? Something relevant, but also simple. Okay. We need to analyze our data. Absolutely. AI and deep learning is the ability to understand how to translate to patients, how to translate, how to box to build models for the in vivo situation. Right now, we have a lot of computer science-driven approaches, but not all of them are really applicable in drug discovery. So real translation is actually necessary as well as experiment design. And probably we can also not just share data that we already have, but we need to design the data that we need to answer a given question, right? If you think back to that pyramid. Okay, yeah, thank you for listening.